Fertilization usually occurs in the ampulla of the fallopian tube. The fertilized egg, referred to as the conceptus at this stage, immediately travels towards the uterus. During the journey, it undergoes several mitotic cell divisions, producing daughter cells called blastomeres. This process is known as cleavage because of the cells divide without growing in volume. After about 3 days, the conceptus arrives in the uterus. It now contains about 16 cells, and it is also called a morula. Cells of the morula are the totifitent. They are capable of differentiating into all cell types, both embryonic and extra-embryonic. The morula continues to divide while floating freely in the uterus for several more days. During this time, it consumes nutrients stored in the egg cytoplasm and uterine milk, secreted by the endometrium. When they are about 100 cells, the cells start to arrange themselves around the fluid-filled cavity, forming a bloctocyst. The bloctocyst consists of pluripotent inner cells mass called the embryoblast, destined to be the embryo, and an outer shell called the trophoblast, which nourishes the embryo. The trophoblast later becomes the chorion, the fetal portion of the placenta. The trophoblast secretes an enzyme that dissolves the membrane surrounding the conceptus, enabling it to hatch, ready to be implanted. At the end of the first week, the blockocyst becomes attached to the endometrium at the site of contact. Superficial cells of the trophoblast fuse together to form the syncytial trophoblast, which grows into the endometrium and derives nutrition from it. In the endometrium, respond by growing over and eventually enveloping the blastocyst, and syncytial trophoblast secretes the hormone named human chorionic gonadotropin (HCG) that instruct the corpus luteum to continue the production of progesterone. Progesterone is the hormone that maintains pregnancy. Among its many functions, progesterone stimulates the growth of nutrients-rich decidual cells to feed the early embryo. Around the middle of week 2, the embryoblast starts to form a two-layered embryonic disc, hyperblast and epiblast, at the midplane of blastocyst with a cavity on either side. These bilinear disc sets are dorsal of ventral axis of the embryo, with the epiblast being the dorsal side. By the end of the second week, the two layers grow around the cavity on their respective side and form the yolk sac and amnion, respectively. The yolk sac provides nutrients absorbed by the trophoblast as well as gas exchange for the embryo during week 2 and 3 before the placenta takes over. Epiblast creating the mesoderm. The remaining epiblast cells form the ectoderm. Each of the three germ layers differentiates to become different organs and tissues. All organ systems are represented in the primitive form by the end of the embryonic period, but most of them are not yet functional or only partly functional. The yolk sac gave rise to the aluntius, a primitive urinary bladder, and part of the umbilical cord. The ambient grows to surround the embryo by week 4. It is filled with amniotic fluid which protects the embryo from trauma and temperature functions. The fluid also allows the fetus to move freely and prevent adhesion of the body parts. The yolk sac, amnion, aluntius, and chorion are the 4 embryonic membranes.
Thank you, everyone! Thank you, Elmar! Boom!